Hello, and thank you, and appreciate the opportunity to talk about ourselves as Ultragenyx Pharmaceutical and talk about our gene therapy pipeline. I'm Erick Crombez. I'm the Chief Medical Officer for our gene therapy and inborn errors of metabolism programs. I've been with our gene therapy programs for the past 7 years, so certainly an exciting time for us to be really shifting into a period of execution as we pull our lead gene therapy programs through clinical development and into pivotal trials. First, a little bit ourselves as an organization. Ultragenyx is focused on developing new treatments for rare disorders, particularly those that have high unmet medical need and sometimes with diseases with no available treatment at all. We are a commercial stage company at this point, with very deep experience within clinical development and commercialization of these products. We have really taken the approach where we've built out various platforms, starting first with enzyme replacement therapy with our lead program, Crysvita, and also then moving to small molecules. Obviously, gene therapy is taking up a large part of our clinical pipeline at this point, but then also very excited about bringing along programs with messenger RNA with our lead program with glycogen storage type 3, and then also focusing on other approaches like antisense oligonucleotides with our Angelman product. And what this does is really provide us with the ability to look at these indications with very high unmet medical need and then figure out the right way to tackle these diseases. A little bit more on our gene therapy programs and how we're executing against them. So we have our two lead programs, our programs for ornithine transcarbamylase, one of the urea cycle defects, and a second program for glycogen storage disorder type 1A, now in pivotal phase 3 programs. We're also excited to be bringing along a program for Wilson disease, a copper transporter defect, into a phase 1, 2, 3 seamless study. So also making that our third pivotal trial in the clinic today. We have moved beyond our initial focus on liver-directed gene therapy and also moving to muscle with our program for DMD, and then into the neurological space for CDKL5 deficiency or CDD. We do also have a program for hemophilia A in partnership with Bayer, and then a very important manufacturing partnership with IHE Sanko. We have spent a lot of time and focus on pharmaceutical development of these gene therapies and manufacturing, and we are excited to be building out a gene therapy manufacturing plant in Massachusetts that will be expected to open next year. A little bit more detail on our three lead gene therapy programs, all of these in or heading into phase 3. Our lead program is an AAV8 liver-directed gene therapy for the treatment of glycogen storage disorder type 1A. This is one of the glycogen storage disorders where patients have lost the ability to break down glycogen to release glucose, which obviously is important during time of fasting for these patients. The current treatment is really based on the use of cornstarch, which is slowly broken down in patients' GI tract, offering access to glucose. And before the introduction of cornstarch in the 1970s, this disease was universally fatal. So it really becomes a disorder of energy management. And we feel that the most direct approach, the best way to restore normal energy metabolism for these patients, is to replace the gene that carries mutations in these patients and restore normal glucose metabolism for these patients. For our primary endpoint for our phase 3 trial, we're looking at reduction of cornstarch while maintaining glucose control. So again, these patients are dependent on cornstarch for life, not only meaning that with missed doses, there's potential for morbidity and even mortality, but also meaning that they need to be taking cornstarch every 3 to 4 hours for the entirety of their life, including overnight. So we're really looking here at the reduction in cornstarch over time after dosing with gene therapy and how they're really able to come off of that cornstarch dependence, which means that they now have the ability to break down glycogen to release glucose during time of fasting. Important also to show that while we're reducing this cornstarch dependence, that we are maintaining good glucose control. And what we're looking at here is how we're measuring that by continuous glucose monitoring using a Dexcom 6 device. The green bars are the amount of time spent in euglycemia or within the normal glucose range, and the blue bar indicating reduction in cornstarch over time. Our phase 3 trial is underway. Again, we are looking at reduction in cornstarch while maintaining glucose control as our primary endpoint, but also looking at other ways to monitor for glucose control beyond the use of continuous glucose monitoring with measurement by finger stick, and then also by direct measurement in blood in a laboratory setting, particularly while we bring these patients into hospital for a controlled fasting challenge to evaluate how long they can fast between cornstarch doses. Our next phase 3 trial is for OTC, ornithine transcarbamylase deficiency. This is one of the urea cycle defects, and this is also an AAV8 liver-directed gene therapy program. So the urea cycle is made up of 6 enzyme 2 transporters. This is one of the proximal defects. It is an X-linked disorder, so it does have a higher incidence of prevalence compared to some of the other urea cycle defects. And for this disorder, it is the inability of patients to normally metabolize protein and handle that breakdown of ammonia, which is a very potent neurotoxin and causing a lot of accumulating neurocognitive damage in patients. So for this phase 1/2 study, we've been looking at patients who have responded to treatment. We're looking at complete responders, those patients who are able to completely discontinue their standard of care. That's the use of alternate pathway medications, the medications that pull ammonia through independent metabolic pathways independent of the urea cycle, and discontinuation of their protein restricted diet, which limits the amount of protein they're taking in, therefore limiting the amount of ammonia that's produced. We enrolled 4 cohorts of patients here and have, across the 11 patients enrolled, 7 patients who have responded to therapy. As an example, this is one of our patients who responded, and response here is defined by a reduction in ammonia scavenging medications and or liberation of diet, but not complete elimination of standard of care. And what we look for over time after dosing with the gene therapy is an increase of that rate of reagenesis. And we do think that assay is very important here, because with the current treatments, the ammonia scavenging drugs, pulling ammonia out of independent pathways, if we can show an increase in the rate of reagenesis, it means we have established normal functioning of the urea cycle and pulling ammonia through the urea cycle for metabolism, and then looking to make sure we're improving or maintaining ammonia control with the data shown in the green line. And then this is a second example. This is what we're seeing here as a complete responder. Again, this is a patient who has completely eliminated their ammonia scavenging drugs and liberalized their diet and are maintaining good ammonia control, as shown here by the green line. And again, looking at rate of ureagenesis increasing from baseline and continuing over time. you do see the decrease in the rate of reagenesis around week 12. We have seen this in other cases and across other programs with the use of steroids. We don't completely understand the mechanism of action there, but we find that we do see some suppression with the use of steroids, but then increasing again once those steroids have been tapered off, and that steroid taper is shown by the gray bars there. The steroid use is really being used across all AAV gene therapy programs as a way to minimize any immune response to the capsid and the gene therapy. Again, it's important for us to really be able to show that we are replacing that deficient enzyme, the OTC enzyme, with this gene therapy and establishing function of the urea cycle. But with all of these programs, it does come back to the patients. How is this really changing their lives and improving their quality of life? And again, this is our second gene therapy program moving into traditional phase 3 trial. This is a very similar design to what we saw with our glycogen storage disorder type 1, looking to enroll 50 patients in a double-blind placebo-controlled trial. Because it takes time to decrease patient standard of care here, we are looking at a little bit of a longer study duration for that efficacy analysis period and looking at 64 weeks of data for that data package. And then again, looking at stability of ammonia while we're reducing alternate pathway medications and liberalizing patients' diets and converting them into responders. And then, again, looking at important changes of quality of life, neurocognitive assessments to show stability and overall rate of hyperammonemic crisis as secondary endpoints. Third program that I wanted to highlight today is our AAV9 liver-directed gene therapy for Wilson disorder. This is a disorder of copper metabolism. These patients lose the ability to normally transport copper. That's the transport of copper through the body for its use as a important cofactor for many important enzymes, but also the excretion of copper out of the body through the bile. With this, the genetically inherited disorder, these patients lose that ability to normally traffic copper, which results with the escape of free copper out of the liver from damaged hepatocytes, and the problems that result of this copper acting in various organ systems as a free radical. Current standard of care is chelation using agents that bind copper to pull it out of the body. That certainly has helped many patients, but still with a lot of unmet medical need here. And again, we feel that if we can directly replace the defective gene, allow the cells to produce normal copper transport that allows for this normal transportation of copper throughout the body, is the best way to tackle this disease. Taking a little bit of a novel approach here, so looking to bring forward a single seamless phase 1, 2, 3 study. We are underway with stage 1 of this development program, and this is trying to move through dose finding, establishing initial safety, and then seamlessly moving into stage 2, which is our pivotal data set. And this allows us to move through seamlessly without stopping the development progress for an end of phase 2 meeting with the FDA or scientific advice with the European regulators. This is a disorder of copper transport, so we will be looking at copper levels in various parts of the body, and then also looking at an assay that allows us to measure the packaging of copper onto protein. And again, really trying to understand our ability to establish the normal trafficking of copper throughout the body. Again, I want to thank you for the opportunity to come and talk to you about us as an organization and to touch a little bit on our lead gene therapy programs in a little more detail. Again, we are a company focused on the development of new treatments for patients with rare disorders and looking at various platforms to do that. Our gene therapy programs are an important part of our development pipeline. And we're very excited to be moving our lead gene therapy programs into pivotal trials. These phase 3 trials are active or soon will be active globally, including within many countries without Europe. So we will be broadening our collaboration and participation with some of these important medical centers throughout Europe, and look forward to enrolling these studies and then being able to share this data in the future with you. Again, this success in the progress with our gene therapy pipeline has allowed us to really innovate and progress on the pharmaceutical development platform, and also invest in a gene therapy manufacturing plant over here in Massachusetts in the United States. So again, appreciate the opportunity to come talk to you about this exciting progress.